Video we will begin with glycolysis. Bismillahirrahmanirrahim. Assalamu alaikum and welcome to Ordinary Days. So, two types of glycolysis are there. One is aerobic glycolysis, other is anaerobic glycolysis. Aerobic glycolysis requires oxygen. Its end products are pyruvate, which can be converted to carbon dioxide and water, but anaerobic glycolysis has end product lactate. The ATP yield of aerobic glycolysis is 36 to 38 ATP, whereas anaerobic is 2 ATP only. The speed of aerobic glycolysis is slow because it has a large number of steps, but anaerobic is very fast because it has a little bit steps. Now, occurrence. The aerobic one occurs in mitochondria, whereas anaerobic one occurs in low oxygen supply cells or tissues. Aerobic glycolysis has byproduct carbon dioxide and water, as we know, and anaerobic has lactate. Now, with the glycolysis thing, glycolysis is like the breakdown of glucose. This breakdown, first of all, requires the glucose to enter into the cell. So, today we will firstly discuss the glucose entry or glucose transport into the cell. It can be sodium ATP independent, it can be sodium or ATP dependent. Firstly, we will discuss this dependent phenomena which occurs in the intestinal cells, renal tubular cells, or choroid plexus of the brain, which has the GLUT1C transporter. Now, this sodium ATP dependent co-transport, it is an energy requiring process. What happens? This is a cell and glucose is outside the cell. So glucose is abundant outside the cell but not inside the cell. And it is sodium is abundant outside the cell but not inside the cell, but glucose over here is abundant inside the cell as well. So in this situation, sodium is going towards its concentration gradient, like it is going from higher concentration outside to lower concentration inside. So the energy over here, it is supplied by sodium. With the sodium, the glucose will also be transported in this reaction, but glucose is going against its concentration gradient. Glucose was more inside the cell. So now to move it against its concentration gradient from low to high, we need certain energy, and that energy is supplied by the sodium, which is moving towards its concentration gradient. So it is a co-type of transport, but this co-transport is done by sodium dependent glucose transporters. Now we have a sodium dependent glucose transporter 2 as well, which is present in kidneys, and it allows glucose reabsorption into the blood. What does it mean? It means that in such a situation, what will happen, these SGLT2, it will allow the glucose to go into the blood. But when there are SGLT2 inhibitors, like in the kidney, like gliflozins, what will they do? They will not allow the glucose to go into the blood. So they will be used to treat hyperglycemia. What is hyperglycemia? It is high amount of glucose into the blood. And how can we stop this hyperglycemia? By inactivating substances that are causing the glucose to go into the cell, go into the blood. And what was causing glucose to go into the blood? SGLT2. So whenever we will activate this SGLT2, glucose will not go into the blood, rather it will stay inside the cell. So it is a treatment of hyperglycemia or high blood glucose in type 2 diabetes patient. I hope you understood. Now in glucose transporter into the cell, which depends upon which is independent of sodium, it is done by the GLUT transporters or glucose transporters, which are 14 in number, but we will discuss firstly 5 ones which are tissue specific. Now 1 and 3, they are present in most tissues. Important one is GLUT4, which is found in muscles and fat, and whenever insulin is more in amount, it will increase this GLUT transporter 4, and this GLUT transporter 4 will actively push the glucose from the blood into the cell. Now this GLUT transporter 2, it is very important because it is present in liver, kidneys, pancreatic, beta cells. And 5-1, it is present in small intestine and testes. So 1 and 3 in most tissues, 4 the insulin dependent one, and liver in the muscles and fat. the two one in liver, kidney, pancreatic beta cells, and the fifth one, which is mostly important, in the testes. Now, this one, the three, which were present in most tissues, and this four, that was insulin dependent, they allow glucose uptake from the blood. So they will allow the glucose to be absorbed from the blood into the cell. But this 5-1 that is present in the testes, it will only transport fructose, not dealing with glucose. But the GLUT2-1 in the liver, kidneys, pancreatic beta cell, it has both activities. So this is a bi-transporter. It can transport both the towards the blood or from the blood into the cell. When there is high amount of insulin, so it will transfer the glucose from the blood into the cell. But when there is high amount of glucagon, like in the fasting state, so it will convert the glucose out into the blood. So GLUT2 has bi or dual activity. Now all these GLUT transporters, the GLUT 14 of them, they transport one glucose at a time, though so they are uniporters. In the next video, we will further start the glycolysis process, that is having two phases: the energy requiring phase and energy generation phase, inshallah.